I think the thing that is super cool is that the elements of SEPUP are a lot of things that traditional publishers are trying to accomplish today, but SEPUP's been doing it forever. So as a result, SEPUP is really good at issue-oriented science, and that makes things relevant for students, that it's problem-based. In addition, it's very student-centered. You're going to see that in looking at the activity we're going to do today, it's easy for students to learn concepts because they're doing hands-on activities to make things concrete. There's a wider range of inquiry approaches, from completely open inquiry to guided inquiry. And in addition, there's support for literacy and assessment. And presently, the third edition is working on being completely aligned with NGSS, but the second edition of SGI is also partially aligned. Again, there's middle school and high school programs, and this is published by Lab-Aids. Just a brief overview of how the program is set up. Good morning, everybody who's popping in right now. It's great to see so many people. Welcome, welcome. As we look at the outline of the course, it starts with a sustainability unit, and every one of the issues that you see that are listed here, that's what makes it relevant to students. So when we look at this, how, especially like ecology, how do you design policies to make fisheries sustainable? So this is introduced very early in the unit, and then it continues to be woven throughout, because the importance is that students are making decisions as stakeholders. And then towards the end of the unit, there's a culminating activity. Welcome, welcome, welcome. I see more people popping in. Awesome. So then they make decisions as stakeholders on to how to solve a problem. So all of these issues that you see are things that students relate to, and it's definitely a problem that we need to solve. All right, so really quick, before we jump into the activity, I think it's important to take just a second, because a lot of times when we work on activities, teachers are always like, hey, wait a minute, you know what, I don't know if my students can do this. And I think the important thing, welcome Alice, welcome, that you understand that this is one activity out of a unit that's in a whole entire program. So as a result, there's things that students have learned previously. This builds on prior knowledge. So before this particular activity in the genetics unit, this activity falls within the genetics unit, students have learned a couple of key concepts. They've learned that individuals all have the same DNA. So we start, the DNA started the same way, but this activity comes right after protein synthesis. So they've also learned that specialized cells make characteristic proteins. So just kind of keep that in mind. Marie has dropped a copy of the student edition in the chat for you to access, and you are going to need that here in just a second. Okay, so really quick, and again, we're not a huge group, so please feel free to either drop in the chat or just turn your mic on, whichever is easiest for you. And the way we're looking at this activity is very much the way we would do this with students. So you really get a feel for some of the questions that are asked and how this activity works in the classroom. So if you take a second, I'm going to post a couple of questions, and it would be great maybe if you have a spare piece of paper. Students would do this in their lab notebook, just to jot down a couple of ideas that you have. How many different kinds of cells do you think there are? And answer these from the perspective of your students. So it's really easy for us as teachers to be like, boom, I know the answer to this. But answer this from a student perspective and try to name 4 different kinds. And then as you think about those cells, how are they similar and different from each other? And then if they all have the same DNA, what makes those cells have different proteins? All right, and again, keep in mind, students just finished protein synthesis, okay? So take a couple of minutes and jot down a couple answers to that, and then we'll discuss this. All right, what ideas do we have? Again, welcome, welcome, either in the chat or turn your mic on. What ideas do we have? How many different kinds of cells do you think there are in the human body? Trillions. Thanks, Donna. Other ideas? We could play the prices right here. Do anyone want to go higher or lower? 250 types. Thanks, Monica. Yay. Any more ideas? Thank you for participating. Hundreds. All right. And again, as we think about this from a student perspective, it kind of gives us an idea, you know, what is it that students think in regards to the human body and how many different types of cells there are? Can somebody else maybe drop in a couple different types? Like, what do you think your students would say if they were asked to name some different types? Whoa, sorry, Darcy, you're going right to AP Bio for some of those great answers. Epithelial, yeah. What else? Anybody else? Type of cell that you think your students would say? Liver cell this year. Okay, they like the liver cell. Blood, nerves, bone, brain. I love all these answers. Great. Neurons. And you know what? It's awesome that you guys are mentioning many of these types of cells, because earlier in the cell biology unit, students have seen blood cells. They've looked at some differentiation, and they have been able to see how different cells have different specialized jobs. And many of the cells that you guys have listed are things that students would be familiar with. So I don't know whether you've looked through the book or you're just lucky, but we've got some great ideas of things that students would have seen earlier. So how would these cells all be similar to each other? Couple ideas. Again, you feel free to turn on your mic, drop something in the chat real quick. They have DNA, they have cell membranes, so good. We're looking at some cell parts, awesome. And how would they be different? Again, thinking about the list you've generated. Function, for sure. Size and shape, great. Thanks, guys. These are great. They have different jobs. I love that you're speaking in student speak, because so many times in workshops, we want to put on our teacher hat and we kind of lose sight of what our kids would say, for sure. So what makes these cells have different proteins then? If they all have the same DNA, how do we get to different proteins? And this is where students struggle, even though we've just done protein synthesis. Any ideas? What do you think your kids would say? Different jobs, different RNA messages. So it's almost like Monica has looked back at activity 16, which was protein synthesis, and hopefully kids would be able to say that. Order of DNA and nucleotides. I love all these answers. These are great. And again, keeping in mind that different genes are turned on. You know what? I would be really happy if my kids would say that, Gina, but I'm not super sure we would quite get to that one at this point. And that's a great segue with what Gina said to kind of step into the activity, because that's exactly what this activity looks at, is how genes express what happens in given situations. So every activity has a challenge question. And just so that you know the challenge question for this particular activity, and again, so there's a copy of the student edition if you just joined that Marie dropped in the chat that you're going to need to access here in just a second. How does the same set of genes direct the activities that all of our different cell types have to accomplish? And so that's the whole idea behind this activity. All right, so to get started, this is what the student pages look like. And what I would like you to do is access the student book that Marie has dropped in the chat. And there is an introduction section. And if you would take just a second and you would read that, and again, kind of maybe highlight or jot it down somewhere, what is gene expression and how does it relate to cell function? So take maybe 2 minutes, read the introduction, and answer those couple of questions. Says you click download. Jordan, I don't know the answer to that question. I'm clicking download really quick to see what happens. Maybe Marie, could you drop the link in the chat so she could access it that way? Oh, a Chromebook, that could be the problem. Hold on just 1 second and Marie will drop a link to the student sheet for you and it'll probably make it a little bit easier to get to where you need to go. Oh, you know what I can do here? Let me do this too. I can open, maybe this will open up for me and since I'm screen sharing too. And that way, those of you who are waiting for the link, I'll put this here. So this is so you guys, for those of you who are having trouble with the link, hopefully you can see my screen. And then I'll move it down here in just a second. And we'll be sure we get that in for you guys, those of you who are struggling. All right, you've got some pictures. I'm going to scroll down a little farther for those of you. It was super short in terms of reading. All right, everybody good on the introduction? I'm going to assume the answer is yes if nobody's yelling in the chat. Yeah, okay, great. All right, so I'm going to go back to the presentation here. If I can move things out of my way. Right. So the thing to understand is that the actual activity has materials. And if you're interested in seeing a sampler, you could swing over to the virtual booth and see if they have some of this to be able to get to you. But so that you can see what this looks like in a normal year, the materials are these chromosomes that the student has out, and they're manipulating silver binder clips and paper clips to kind of work through this activity. So in a normal year, students would read the activity, and then we would do the procedure steps. And so they would do this, using their textbook. And I'm going to warn you, once we jump into the Jamboard, you're going to really have to read the directions carefully, or this won't work for you. We have made it a little bit easier with the Jamboard, but still yet you want to make sure that you are looking carefully at that. So Marie's going to drop a copy of the Jamboard into the chat for you. And this should work even if you're on a Chromebook. Shouldn't be a problem. Are we getting the Jamboard?
I put it in the chat.
Awesome, thank you. All right, so once you're in the Jamboard, and I'm going to go ahead and pull it open so you can take a look. Hopefully it'll open for me too. You're going to look at the instructions.
It's saying that they need access to get to it. Should I try the other URL?
They should just be able to copy the document. Hold on, let me. Not quite sure why it's not working for them.
Try the tiny URL.
Yeah, maybe try the tiny URL.
I just dropped that one.
And see if that works for you guys. Tiny URL's not working either. What is happening? Why is this not working? Hold on just a second. Let me try one more thing. Because I know we just used this. Paste. Whoops, let me try this. All right. Try that one, see if that works for you guys. And then someone, yay! Oh, I am the most tech awful person on the face of the earth, and the fact that I just got that working is amazing. And I didn't sweat through my shirt either, just so you guys know that. Whew. All right, so go ahead and open up your Jamboard. And again, you're going to look at the first 6 steps. And as you work through this, follow the procedure. And for the sake of time, once you get to the 3rd one, you are only going to look at red blood cells. So I'm kind of paging through this so you can kind of see what you're doing. And then once you get to procedure step 6, just stop. So I'll give you a couple of minutes to do that. Donna, let me repaste and see, maybe, Donna, did you try this link? I'm not getting any requests to access it. Yeah, just go ahead and make a copy, Donna. And then once you hit make a copy, it may be a little slow to open for you. But let me know once you get in. Oh, yay. You're welcome. And again, if you are not sure as you're working on doing this, please just turn on your mic or drop something in the chat. Marie or I will catch it. All right, finally in. All right. Oh no, sorry, Michelle. Give you about 2 more minutes. About 1 more minute. Awesome, Candace. I am glad that, oh, so SEPUP, are you in middle school, Candace? High school, great. And you use biology? And Erin, that's okay. It'll load. We'll talk through it. Fantastic. Yay. Erin, it's okay. It'll load for you eventually. And you won't lose access to the link once this ends, so you'll still be able to use it. And yeah, for all of you who are struggling, Michelle, Michelle, do you need the link for the Jamboard? There we go. All right, so let's take a look. And I know some of you are still waiting for Jamboard to the link for the activity. So you should be in pretty good shape. Michelle, if you have the Jamboard, everything in the Jamboard pretty much has the activity. And then Marie, is there a way to reload the stuff like the student sheets and for people?
Yeah, I can resend the student edition and the teacher edition.
Perfect. Perfect. All right, so let's take a second and chat about part A. And basically what you guys saw were your 2 chromosomes. And you had to go through and make some marks on those. Which of those genes were active? So think about these questions and kind of look at what you have on your Jamboard. You'll have to go back and do a comparison. Which of the genes are active in all 4 cells and why? Which are active in only some of the cells and why? And which protein isn't made for any of the 4 cells and why? So again, you'll have to look at your Jamboard, and if you don't, and if you don't have the Jamboard, let me pull it up for you so you can see it.
I'm going to give you a quick 15.
Thank you. So this is what it looks like if you have not been able to access the Jamboard. All right, and heading back to those questions that we had. My internet's slowing down too. We're having a wonderful storm right now, so I'm hoping my internet stays with us.
Yeah, we lost power.
Yeah, I'm afraid. All right, so as we look at these questions, were there genes that were active in all 4 cells? Anybody? Oh, are you all ghosting me now? You're all starting to act like students. You were all enthusiastic and now you're like, oh, I'm not going to talk to her. Yeah, there were. So even if we don't want to name those genes, even if the cell growth controller. So let's just talk about that one really quick. Why do you think that gene would have to be active? Why do you think the cell respiration enzyme? Why do you think those would have to be active? Thank you guys for dropping that in there. Yeah, we need ATP, we need energy. And again, kids would know these things from previous activities. We don't want all cells to grow wrong. Good, these are great answers. And so these would be just great. We're not going to go through every question. So we definitely we would have a stopping point to stop and talk with kids about this so that they would have an idea. Why do certain things need to be in every single cell, and why are there only some in some cells and some of them they're not in any of them? So this brings up a good discussion point, but also keep in mind that these things are based on content that students have learned previously, but they're also things that require them to make good observations too. They have to be able to look at that chromosome chart. They're sharing that information with each other, and they're coming up with conclusions just like scientists do. All right, so I know that this is going to be quick because I don't want to keep you guys over your time. But in the Jamboard, you're going to go back to that, and you're going to do steps 7 through 12. Now, this is what this looks like in a real year. You can see that you've got all these paper clips on here. But in the Jamboard, I'll go back to that if I can get out of this really quick. Whoops. Sorry, I'm having trouble navigating now. In order to do this, this has gotten real super slick. Again, you may want to just jot down one or two of these, but on the 9th slide of the Jamboard, now you start looking at cellular events. And in order to do that, you will have to click on the top card, and it will allow you to reverse and see what's on the back side of it. So take a couple of minutes and just kind of click around on maybe 2 cellular events, maybe 3 if you have time. And then based on the information that's on the card, you're going to do step 9 and then move forward to frame 10. So take a couple of minutes. And I know some of you are still struggling with the Jamboard. And if you are, let us know. Watching my clock here. Then when you guys flip over, you'll be able to move your paper clips. You should be able to drag and drop them wherever you need to. Take about 1 more minute. All right. So I know you may want to play some more, but again, you have this Jamboard, so you can play as much as you want. But for the sake of time, we want to go back and just from looking at the couple of things that you did, were there any changes or anything that you saw in regards to the environment that led to short-term changes in gene expression? Anything you ran across?
I'm going to give you a quick 5-minute warning.
Thank you. Anything that you saw? You have, that's okay. That'll be your answer, Michelle. Odd internet. That gave you short-term changes to your gene expression. That's what we're going to go with for an answer for that, for you. Anybody else? Did you get that far? Giving you a second. No one wants to hang out with me here. You're ready to move on. Yeah, I saw someone nod their head. So you may have not flipped the right cards over, but certain cards give you a short term, like for instance, if you eat a heavy meal, one of the cards in the event cards talks about eating a heavy meal and how that affects, the certain digestion of things and how that leads to a change short term in gene expression. So this allows students to kind of explore what happens that would actually change what we see in terms of their genes being expressed. On the marketplace asking. Perfect. Nope, you're fine, Monica. Do that. This lab is wonderful. It does a great job of showing kids, in a very hands-on way what this looks like. You got that a lot of hemoglobin would have to be produced quickly. Yeah, that would so then we would have to go in and we'd have to repress that. Yeah, absolutely. Thanks, Alice. And it really all boils down to whatever card you flipped over in this to answer this. But notice that in the table that you saw, this had students do this like 6 times. So they're going to see multiple different things that happen, and they're going to talk about it with their group because everybody has different type of cell that they're looking at and how it affects that cell. So in the beginning, I mentioned real world connections. And as part C for this activity, students do a reading about GMOs, and the genetics theme or the genetics issue is genetically modified organisms. And they look at how that's controlled with gene expression. There is a literacy component that supports students in terms of doing the reading. So that's always helpful for our students who need that assistance. Just a general overview, once again, of each of the content that you find in each of the units, roughly how long it takes to complete it. You notice this kind of nicely breaks out into quarters when you add it all up. And the thing that's really nice about this as a teacher is how this stuff is packaged. When you buy SGI Bio, it comes on these wonderful carts with these boxes, and pretty much everything you need is in the box. You are not going to be Walmarted to death by this particular program. And if you do need something, say for instance, beans, it'll tell you that in the teacher's guide. So it makes it super easy for you to find the things that you need. This just kind of gives you a quick look on the inside. There's enough materials to carry this out for 5 classes of 32 students. So that's quite a few students that you can work through with these materials. The lab notebooks are wonderful. All the things that we talked about in regards to the lab notebook, students would have put in their notebook. So you can see some examples of student work here. More examples of literacy to help support students not only in reading like you saw earlier, but also in writing. There are not only formative checks throughout every activity, but there are also bigger checks that are that scoring guides are provided for. They have multiple scoring guides that can be used throughout the program to support where students are, and you get a feel, just a pulse for where students are. Jessica, let me see. In our district has purchased Lab-Aids. Do you have these virtual lessons available? Jessica, that's a great question. If your district has purchased any of the year-long programs, some of these are available through the portal. This particular lesson was made virtually as, again, Lab-Aids moves towards being able to do things like this. However, there's a couple that they haven't gotten to quite yet that I've just made Jamboards for and moved forward. So Jessica, it really depends on where your level of purchase was, whether it was for a full year program or for an individual kit. This is the portal. I'm glad, Jessica, you asked that question, because this is the online, so you can have access to curriculum materials, the teacher's guides. This is what this would look like if you purchase materials. Another thing that's really nice as we've moved into this virtual world, and Jessica, I'm glad you brought that up, is that Lab-Aids for some of the labs have also developed what are called lab sent sheets and videos. So really quickly, I'll just give you, this gives you a couple examples of what that looks like for students. Again, if this will work, because my internet's dragging on me. Come on. Oh, there we go. Maybe. And this is a different activity where students don't have the opportunity to touch those materials. They don't have the opportunity like this year to make slides. So the slides are made for them, and then their images that they watch. And then there are other remote learning resources that Lab-Aids has put together, not only for SGI Bio, but some of their other programs as well. So please feel free, if you have your phone, you want to screenshot that real quick, you're welcome to. Full year programs have professional development, which support them, and their professional development is great. And if you want more information or you want to request a sample, you can go to the website, or you can swing by the virtual booth and see what they have available. I can't promise that there's a sampler available for everybody for this activity today, but usually they're pretty good about having excess that you might be able to get a hold of. And I'm trying to get back to the poll. I'm going to throw the poll up. So I'm going to launch this, and if you guys would be kind enough to just do the poll really quick. And if you have any questions, I'll leave the chat on, and you're more than welcome to drop them in the chat, and I'll stay and answer them. Or you can just turn your mic on and ask questions too. I'd be more than happy to answer questions. You're welcome, Jessica. The portal is great. So definitely, and Jessica, let me give you this, Jessica. If you're using the biology program, here's my email. Feel free to reach out, and I'll be happy to try to help you however I can. Thanks, Michelle. Have a good day. Enjoy the rest of your conference.